What is up? Ready for the first test of the new 9 weeks? Yeah, I know I am. So, first and foremost, there's a outside chance. I don't know what kind of chance it is, but there's a small chance might not be there tomorrow. So, one of my kiddos this evening has a little fever. We're gonna have to kind of play it by ear if he's going to be able to go to school in the morning. And if he can't, then I'm going to have to stay home with him. So you'd have a sub, you'd have Miss Cordaway there. Again, it's just a test day, so it's really not that huge of an issue. Besides, if you were planning on coming in for tutoring, if that is the case, then definitely shoot me an email with any of your questions or a remind message of any questions you might have or things that you weren't clear about. And then hopefully this video will help as well. But again, hopefully I'm there. Hopefully he's good to go in the morning, and I'll be able to be there. So we'll have to see. So let's just kind of start with going over all of our stuff that we've been talking about since we've been back. So we started with this whole idea of protein synthesis, right? So which I said was basically how our DNA, so all the DNA in our cells, it holds the instructions on how to build proteins, which then in turn build us. But remember, there was that issue. DNA is found in the nucleus, but proteins are built in the ribosome. So we have to get the instructions or the blueprints from DNA to the ribosome, where the protein's actually going to be put together, where the instructions are going to be read, where the correct ingredients are going to be put together to build the protein. And so there's a couple players. So we have messenger RNA, okay, and that's what it sounds like. It takes DNA's message out of the nucleus to the ribosome, and that process where DNA gives the message of the blueprints of DNA to RNA, that's called transcription. And remember, you just use your base pairing rules. So A to U, T to A, G to C, C to G. So the RNA base pairing is very similar to when we did DNA base pairing, except remember, RNA doesn't have any T's. It has U's. It has uracil instead of thymine. So that's step one, transcription. DNA gives its message to messenger RNA. It's going to take that message out of the nucleus and go to the ribosome. Most ribosomes are found in cytoplasm, and so you might see, it's the protein's being built in the cytoplasm. Remember, specifically it's at the ribosome, but ribosomes are in the cytoplasm. So then the next step would be what we call translation. So translation is when we take chunks of the messenger RNA, and the ribosome reads those chunks and puts together the correct amino acids depending on what it's reading. And remember, when we say chunks of messenger RNA, we're talking about 3 bases. So 3 bases of messenger RNA are called a codon. So codons code for specific amino acids. And remember your codon chart. You'll have to be able to read those, but you'll look at your codon chart and it will tell you then what codon is going with the certain amino acids. So that's step 2. So translation, you're taking the messenger RNA, and the ribosome is going to read that and determine what amino acids are needed by looking at those codons. The other major player in this is transfer RNA. So transfer RNA, again, kind of like the name sounds, it's going to transfer the amino acid to the correct spot on the messenger RNA. And we talked about in class how if RNA has codons, which is the set of 3 bases, transfer RNA has the anticodons that are going to match up to know where to bring the amino acid. So again, if we equated this like I used in class, kind of the recluse architect. So DNA is kind of the architect that's a recluse and stays in his office and never leaves. And so he has all these great blueprints, but he needs to get them to the job site. So messenger RNA is going to be that intermediary that's going to take the blueprints from DNA to the job site. So that's transcription. And then once messenger RNA is at the job site, they're going to call over and kind of get all the materials in the right order. And so the workers that would be bringing the materials would be the transfer RNA. The materials themselves, the lumber, the nails, the screws, everything that you're going to build a house with, those would be the amino acids that we would put together to then build the protein. And then the final product, like we said, would the house or the building or whatever you're actually making and creating, that would be equivalent to the protein. So there's just kind of the little story that we told that goes along with our protein synthesis process. The next thing we looked at mutations. And remember, mutations is just any change in the DNA strand. So mutations occur to the DNA. And then, as we talked about today in the review, the result of a mutation can be a change in the protein that's formed, because as that DNA gets passed on to messenger RNA, if the message isn't what it originally was, then it could code for a protein that it really shouldn't be making. Or it might code for no protein at all. As we saw with some of our activities, the protein can get stopped short and not fully function. So that was the mutations, and there was a couple different types. So we had a point mutation, which only affects one amino acid, or really only one base on the DNA. So point mutation would be, an example would be a substitution. So let's say you have an A in the original strand of DNA. Well, it got flipped and a T substituted in. So now instead of an A, there's a T. So that would be a substitution and a point mutation, just kind of the example there. The other type, the kind of big category we talked about, were frame shift mutations. So that's, remember in class when I had people holding up the boards, and then I took one out and shifted them over and showed you how it kind of messed up the whole DNA strand. Those are frame shifts. So deletion and insertion, those would be the two different types of mutations where it's going to be messing up more than just one codon. So, and again, those are called frame shifts. As I said before, on the test, you need to just be able to make sure you can identify a substitution or a deletion or an insertion, but then also be able to, okay, here's the original strand of DNA. What is the amino acids that's going to code for, and then if here's a mutated strand, what new amino acids. And really that shouldn't be a problem as long as you remember DNA to RNA, and then you find your amino acids with the chart. And that's really all there is to it. The last part we talked a little bit about it today was gene expression. Remember, the big thing, the big idea is the environment can affect how genes are expressed. We talked about, in some class, we talked about there's bacteria that when lactose is present, then the bacteria create the protein, and that's the environment. That environment of lactose is making those bacteria create the protein. Other classes, a little bit easier of an example, we talked about the Arctic fox, right? That was in the video. So the Arctic fox has white hair in the winter and dark hair in the summer. And again, it's not like the fox is wanting to change its hair. The temperature is affecting those genes, and the genes are expressed differently depending on the temperature. So the environment is affecting the gene expression. Okay. All right. So we also did a little mini lesson on biotechnology. We talked about like DNA fingerprinting, how you could compare to see paternity tests, or you could use it in crime scenes. But basically, you can compare different organisms, not within the same organism, to see similarities in their DNA by using DNA fingerprinting. So go through now and just kind of look through some of the questions and make sure we've covered everything. Kind of give you an outline of what we've got. One of the questions dealing with DNA fingerprinting, just kind of asking you kind of what you can do with it, which we've talked about. What you're going to need, what's the monomer Nucleic acids, need to make sure we know that. What's the whole goal of, what is DNA even for? And that's what we've talked about. It's for making proteins. It's this whole idea of protein synthesis. The DNA has the instructions on how to build proteins. That's DNA's main job. Remember, messenger RNA is single-stranded. Messenger RNA helps to carry DNA's message out of the nucleus. Yeah. Yeah, like I said, know the nucleic acids. The monomers of a nucleic acid is a nucleotide. You're going to have to look at models and tell, is that transcription, is that translation, kind of like we did with Kahoot today. And again, you just need to look for the certain clues to let you know what's going on. There's a mutation question, just asking kind of what's the difference. Shouldn't be too hard. Using a codon chart with mutations like we talked about. There is one karyotype question, I will tell you that. You need to just look at the karyotype very, very carefully. Remember, the karyotype's a picture of a person's chromosomes, and if there's an extra chromosome, if there's a missing chromosome, then that's going to be an issue. Remember, if it's XX, it's a female. If it's XY, it's a male. So at the 23rd spot, if there's two matching ones, that's going to be a female. At the 23rd spot, if it doesn't match, if it's like a big one and a little one, then that would be a male. But again, you just need to look at the karyotype carefully and make sure you see whether there's an abnormality. You just gotta read it careful. More diagrams. Again, looking at transcription or translation. You're going to need to put the steps of protein synthesis in order. So, DNA gives its message to messenger RNA. Messenger RNA goes out to the cytoplasm, to the ribosome, to help to build the protein. So, all of that stuff. Transfer RNA brings the amino acids. So, you'll just need to put the steps in order. Let's see. Gene expression. Again, we've talked about that today. We've talked about, with mutations, they're not always a bad thing, right? I think I mentioned this in class when we get into evolution a little bit and we talk about variation within a species. Variation wouldn't exist if we didn't have mutations. If there weren't, little changes in DNA here or there, there wouldn't be variety within a species, which, again, that's a good thing, especially when you're talking about natural selection and survival of the fittest. We wouldn't have organisms that were fit if there weren't changes in DNA that were kind of benefiting them and creating diversity within a species. So mutations aren't always a bad thing. What's another codon chart? Just again, got to kind of be able to read it. More looking at being able to do translation, almost kind of backwards. Like they might give you some amino acids, and then you have to say, okay, what codons did these come from, or what could have been the possible line of codons. So that shouldn't be too hard. I think really there's not a lot of. You just got to read careful. I think you guys do a good job of that most of the time. I'm not seeing, again, a couple models at the end, a couple more pictures to interpret. Again, just saying what's going on. Is this looking at transcription? Is this translation? Remember, your biggest clue there is going to be if there are anticodons involved, if there's transfer RNA involved, that's going to be translation. Also, if it's happening at the ribosome, it's going to be translation. If there's DNA involved at all, it's going to be transcription, because that's when DNA gives the message over to RNA. So I think this has been long enough, but hopefully I covered everything, and and got everything you need. Especially, like I said, if I'm not going to be there in the morning, I know stuff happens with 4 kiddos. Things happen. Hopefully, it'll work out. But I will send a remind out kind of one way or the other in the morning. I hope y'all have a good night. Like I said, let me know if you have any questions. Shoot me an email, send me a remind message, and I will do my best to get back with you and answer any questions you might have. Y'all have a good night. We'll see you later.